Hey, good morning, members. Thanks so much for joining us today for a member talk all about the history of the Academy in the Galapagos Islands and how specimens collected there over 100 years ago are being used to further current scientific work. Joining us today is Curator of Ornithology and Mammalogy, Dr. Jack Dumbacher, who I'll bring on in just a second after some short housekeeping notes. So we'll be taking questions from you after Jack finishes his talk. Please go ahead and submit any questions you have in the chat box to your right during the talk or after the talk, whenever the question comes to you. If you need any help setting up a YouTube account in order to comment, just scroll down in the YouTube description. We have step-by-step instructions there. So without further ado, let's bring Dr. Jack Dumbacher to the screen. Hey, Jack, good morning.
Thank you. Good morning. How are you?
Very good. Well, I think I have your presentation here, and so are you ready to get started?
I am.
Okay, excellent. Thank you.
Go
See
for
you
it.
on the other side.
All right. Thank you. Thanks for joining today, too. I want to talk a little bit about the California Academy in the Galapagos, a century of work on birds. And let's start with, what are the Galapagos and what do we think of them today? And so I bet you already have some ideas of, your impressions of the Galapagos. So feel free to put them in the chat. I'm kind of curious what you think of the Galapagos. But I'm going to bet that you probably know a little bit about it. It's an island archipelago that's about 1,000 kilometers off the coast of Ecuador, off of South America in the Pacific Ocean. And there's 12 to 15 larger islands, major islands, and then a whole bunch of smaller islands going down to, basically the size of rocks. And you might think of, beautiful sandy beaches and barren volcanic cones. And you might think of, in the high Scalesia forests on some of the taller islands that are lush and green. But one of the things that the Galapagos is really known for is the wildlife and the opportunities to get really close to the wildlife and see the wildlife in beautiful ways because it's so tame, and partially because there have been, so few people on the Galapagos for so long. And they're so tame and so abundant that there's often signs telling you to keep away from the wildlife or give the wildlife a little bit more space. And this allows, tourists and folks who visit Galapagos to see wildlife in a really amazing way. So you can get up close, you can get amazing views of things, great photographs. And so here are a few of the things you might see, like the swallowtail gull, an American oyster catcher. This is a red-billed tropic bird, which breeds on the islands. The marine iguanas, which you find on virtually every island and even some of the small rocks, are really very fascinating animals, and they come right up onto the roads in the towns. The waved albatross, most of the world's population breeds on the Galapagos on Espanola Island, beautiful large birds. Sully lightfoot crabs are all over the intertidal zones and on the dark rocks. Galapagos has its own flamingo, and here's a picture of it. And blue-footed boobies are also iconic for the Galapagos. But then there's also some really interesting things that tell us about evolution. So here's the flightless cormorant, a cormorant that got out to the island, and then its wings became so reduced that it couldn't fly, but it can swim very, very well. And then, of course, the herpetofauna is really interesting. I've already mentioned the marine iguanas, but they're also the small lava lizards that you see in the upper left and lower right, and then land iguanas. And these have all evolved some differences on each of the islands. And of course, the Galapagos finches, aka Darwin's finches, that have become almost synonymous with evolution. But it wasn't always this way. And so how did the Galapagos become this iconic place for the study of evolution and for understanding evolution and for naturalists to go and visit? So now we go back to the voyage of the HMS Beagle and Charles Darwin, who was the young naturalist aboard the Beagle, traveling around the world to document natural history and study. And he wrote to his cousin from Peru in 1835 that he looks forward to the Galapagos with more interest than any other part of the journey. And with all the interesting places he went, it still blows my mind the Galapagos was one of the ones that he was most interested in. But the Galapagos did not disappoint Darwin. So Darwin and the Beagle spent about 5 weeks in the Galapagos in the late 1835, and they only actually visited 4 islands. And these are Chatham Island or San Cristobal, Charles Island or Floreana, Albemarle Island, or Isabella, and the last one that they visited was James Island, or Santiago. And as they were on the islands, they were told by some of the Spanish who lived there that if you could bring me any tortoise that you collected from any island, that they could tell Darwin which island that tortoise came from. And you can see how the shell differs. This is a dome-shaped tortoise, and this is more of a saddleback tortoise with a higher flange on the front and a longer neck. And Darwin saw this as he traveled from island to island, and he could see the differences in the shells. But even more than the tortoises, which he was told about in advance, were the Galapagos mockingbirds. And each of the islands that he visited had a mockingbird that looked different. But on the island, they didn't seem to vary very much. But between the islands, or among the islands, they differed incredibly. And so Darwin actually was able to collect some of these mockingbirds and take them with him on his voyage. And so he had time to muse over them and think about what was going on. And so he wrote on the mockingbirds of Galapagos, he wrote, "I have 4 specimens from as many islands, and there will be found to be 2 or 3 varieties." In fact, there turned out to be 4. And each variety is constant on its own island. And this is a parallel fact to the one mentioned about the tortoises. He goes on to say that, "When I recollect the fact that from the form of the body, the shape of the scales, and general size, the Spaniards could at once pronounce from which island any tortoise may have been brought. When I see these islands inside of each other and possessed of but a scanty stock of animals tenanted by these birds, but slightly differing in structure and filling the same place in nature, I must suspect that they are only varieties. The only fact of a similar kind to which I am aware is the constant asserted difference between the wolf-like fox of East and West Falkland Islands." And here's the kicker. He finishes with, "If there is the slightest foundation for these remarks, the zoology of archipelagos will be well worth examining, for such facts would undermine the stability of species." So in that one sentence, not only does he understand that species may be evolving or changing or mutable, but he also realizes that archipelagos, that islands, are a natural laboratory for studying evolution. And so this insight led to the study of islands and the study of looking at evolution across islands. So in this light, the Academy in the late 1800s was reading Darwin, thinking about evolution, and visiting islands of its own, and became interested in the Galapagos. So remember that the Academy was founded in 1853, and its charter was a thorough, systematic survey of every portion of the state and the collection of a cabinet of her rare and rich productions. And by 1891, Cass had a beautiful museum in downtown San Francisco on Market Street, shown here on the left. And it was expanding its work into the Pacific, and it had led trips out to the Channel Islands in Southern California and the Revillagigedo Islands off of Mexico, and their aspirations were growing to survey other parts of the Pacific. And they sought to compete with the large academic museums in the eastern United States. Now, Cass curator and director Leverett Mills Loomis in 1902, 1903, began to be interested in a big multidisciplinary expedition to the Galapagos. And to lead this expedition, he chose Rollo Beck. And Rollo Beck had already been to the Galapagos on multiple occasions with Lord Rothschild from England. And on one of those expeditions, he took this photo. And at the time, even though it was well known that there were very interesting tortoises on the islands, they were mostly being hunted by whaling ships and pirate ships and buccaneers and all the people who visited Galapagos, because they could be used for easy meat. And so they could be chopped up right on the islands and eaten, or they could be thrown on board a ship and be kept fresh for many, many weeks so that sailors had meat to eat. And because of that, Rothschilds made a sad prophecy that the tortoises would likely be extirpated in a matter of years. And this was a major motivation for the expedition, because people realized that the islands were changing. And many of the things that Darwin was interested in which told us about evolution, were likely to disappear unless they went and they collected them, so that we would have the natural history record in a museum. And so because of this was part of the motivation for that expedition. And so Loomis amassed 8 young scientists, and also a navigator and a cook. There was no captain because Beck was in charge of the expedition, even though he wasn't in charge of the vessel. And we had a botanist. In fact, we had a botanist, we had herpetologists. This is Beck in charge of birds. This is Hunter, who collected birds and also mammals. This is Parker, the navigator. This is Slevin, who was in charge of herpetology. This is Gifford, who was only 17 years old at the time the ship sailed. And then this is Oxter, the geologist, and Williams, the entomologist. And then this is also Nelson, and he was the cook and the first mate. In order to make the trip, they purchased a 85-foot schooner that they renamed the Academy. And it was previously a U.S. military ship that had run aground on Angel Island. So they had to pay for a lot of repairs and get all the rigging redone. But they were able to reset the boat and send it to the Galapagos. It stopped along the way in a couple of islands off of Mexico, the Cocos Islands, and then it spent a year and a day in the Galapagos. And here you can see the path that it took around the Galapagos, visiting many different islands, and some of the islands multiple times. And even most of the smaller islands were visited and sampled and collected. Now, on the islands, these men went to every little corner, every cave, every crevice. They were able to collect fossils, insects in the caves. They collected tortoises and carried them back to the ship. Here they are studying nests of birds on the Nazca boobies, pulling marine iguanas off of every rock they could find, putting their arms into the burrows of petrels, and they even found Galapagos penguins on the islands and were able to bring those aboard. They sampled the eggs and nests of birds, and because they were able to revisit many of the different islands, they were able to sample them in different seasons and understand how the birds changed from season to season and which ones went out to sea and which ones stayed on land. They also had a camera, and this is Rollo Beck taking some cameras. And so they were able to document all the work that was done. They probably had at least 2 cameras or they wouldn't have been able to get this photograph. And so they were able to document the work that they did and the places that they went, what the habitat looked like at the time, and how they were able to accomplish all the work that they did. And not only did they make amazing natural history specimens that will be at the Academy for a very, very long time, documenting what they found, but they also took copious field notes. And here's an example of some of Gifford's field notebooks, painting pictures of the birds, making maps of where they went, with lots of information about the behavior of the animals that they surveyed and found as well. Now, while they were on the islands in 1905 and 1906, disaster struck San Francisco in the form of an earthquake and fire in April 1906. Now, this destroyed the museum. So the building was burned, and all of the contents, except for a few specimens that Alice Eastwood was able to pull out, primarily in the botany collection, were all lost. And of course, when the folks on the ship heard of this, they said, they knew that there was trouble. Then they wanted to go home and be with their families and help restore the city. And Loomis realized that this was going to happen. So every day he sent a letter, hoping one of them would eventually get to the men on the ship, telling them that you must stay on the ship. Your family's fine. We're doing everything that we can. We don't need your help. We need you to stay in the Galapagos and continue the collecting. And so that was when J.S. Hunter heard about this, and he stood on the boat clad in his tropical costume, and he said, "We are the Academy collection now." They were all that was left. So it was so important for them to continue the work. They returned to San Francisco on November 29th, 1906, Thanksgiving Day, with 28,000 specimens on board. And these were split up with 38,000 shells, 1,000 invertebrate fossils, 13,000 insect specimens, 10,000 plants, 5,000 reptiles, 8,688 bird specimens, of which 3,800 were specimens of Galapagos finches, 120 mammal specimens, and 2,000 eggs. So it was a huge collection, 78,000 specimens. And of course, there was no place to put them. So they had to be stored on the ship for a few weeks, probably even months, until they were able to find a space that they could store in. So here's the cast tortoise collection in its temporary holding place until they were able to rebuild the museum in its permanent place which reopened in 1916 in Golden Gate Park, and then eventually was built out to all these different spots that you see here. And then, of course, it was rebuilt in 2000, between 2004 and 2008, the academy that we know and love today. So then the question is, well, how has this expedition to the Galapagos and all the specimens that were collected there, how has it altered our understanding of evolution? Well, I'm going to talk primarily about birds because that's what I know. I'm the curator of birds and mammals at the California Academy of Sciences, but I'll tell you that every collection that we have at the California Academy has its own story to tell. So I'm going to be telling the story of birds. But when the specimens came back, there were many different monographs published about, of course, by Leverett Mills Loomis on the Procellariforms, the petrels, the storm petrels. But then all of this came together when Harry Swarth became the curator of ornithology, went through all the specimens, and he wrote a thick monograph called the Avifauna of the Galapagos Island. And in that volume, he laid out all the species of finches and seabirds and other land birds, basically as we know it today, with very few changes that have happened since, mostly due to modern genetics we have now. And the Galapagos finches were especially difficult to sort out. In the late 1800s, some other researchers, like Robert Ridgway at the Smithsonian Institution, believed that there was really only one species of Galapagos finch, because with a lot of variation, because when you line all the specimens up, you could find a perfect gradation from those with the smallest bill to those with the largest bill. But because of Swarth's work, he was able to show that they actually come in clusters, and so that you can tell the large-billed groundfinch from the medium-billed groundfinch from the large groundfinch. And so he was able to do this and basically come up with the species delineation we use more or less today. Now, after that came David Lack, and he wrote this classic volume that came out in 1945 on numerical taxonomy. And he was especially interested in the Galapagos finches. Now, we already mentioned the tortoises and how each island has its different form, but the Galapagos finches were extremely complicated because each island would have multiple finches. And so trying to understand what had happened and how they all came together was incredibly complicated. So Lack had measured literally thousands of specimens of finches from the Academy collection, also from the Stanford collection, and was able to put all these together in order to understand the evolution of finches. But he wrote in that 1945 volume, in fact, he quotes Darwin. And one of the interesting quotes that Darwin offers, he says, "Whereas it cannot be disputed that the nature of other species with which each has to compete is at least as important and generally a far more important element of success." But afterwards, Lack goes on to say that there is no evidence of Darwin's suggestion. And in fact, there is no evidence whatever in any of the island forms of Geospizinae, which are the Galapagos finches, that their differences have any adaptive significance. So he believed in this first volume that adaptation had nothing to do with what was going on with the Galapagos finches, which is very interesting and, of course, is not the final word. So he actually submitted this manuscript shortly before World War II. And then because of World War II, there were delays in the publication. It came out in 1945. But during that period, during the war, Lack continued to think about this, and he continued to look over the numbers, and he continued to talk with friends of his, and he totally turned and changed his story completely, and he republished it in this book called Darwin's Finches, which came out in 1947. And in this volume, he was able to clearly show exactly what Darwin had suggested earlier, that the different species are each other's most important evolutionary force. And so if you look at any one given island, you might find that the small-beaked groundfinch, has this size, and the medium groundfinch has this size, and the large groundfinch, has bills of this size. But when the large groundfinch isn't present, then the medium groundfinch might be a little bit larger. And when only one species present, for example, the medium groundfinch is the only species present on Daphne, then it'll shift in order to take advantage of the different seeds and the things that are there. And likewise on Los Hermanos, only the small beaked groundfinch is present, but its bills are about the same size as those of the medium groundfinch on Daphne. And his conclusion is that what's happening is that as the different species are competing, that they're actually sort of agreeing evolutionarily, or their adaptations are sort of parsing up the resources so that they all can compete and exist on one island. So this has become classic work on community ecology and has actually kicked off the field of community ecology. Incredibly important work. And then after Lack's work, Bob Bowman, who was a professor at San Francisco State University, went on to further study Darwin's finches and looked at exactly what seeds were available on each of the islands, and was able to further understand why the finches' beaks are the sizes that they are, and why the different species are able to exist on each island based on the food that's available, and how each of the finches is splitting up the food, the plant food and the animal food, and having different types of bills to probe or pick or cut or crack different seeds, and how they've actually adapted to the different environments on the different islands. And this too has become classic. And these charts of Darwin's finches' bills are classic things that you see all over in the Galapagos in every gift shop. And of course, one can't forget Peter and Rosemary Grant. And even though they didn't use the Academy collections as much as many of the others, they worked on the islands and did a lot of work to show that evolution actually happens in real time. So for a long time, we thought, well, evolution happens, but it happens over millions of years, and you can never witness it happening because it happens so slow. But they worked on Daphne major, and in 1976, they had surveyed and measured every one of the 751 finches on the island, and were able to look at the bill sizes of the finches on the island. And then a severe drought happened in 1977. And after the drought in 1978, they also remeasured the finches and could see the shift in the mean, that the bills had become larger, and were able to understand the seeds that were on the island and how the beaks actually had adapted to the changing availability of food and in response to the drought. And interestingly, just as we would believe from evolution, of course, the beaks remained larger because they had actually evolved. It wasn't just a short-term thing that had happened. There were actually gene frequency differences. And so a lot of our understanding of how evolution happens has been done on the Galapagos, and a lot of it's been done with the Galapagos specimens at the Academy. So then the question is, well, do we still use our Galapagos specimens for science? And of course, because I'm here, the answer is yes. And again, there are so many different researchers that use our Galapagos collections. It's really, part of the foundation of our collections. So I'm going to give you some examples of some of our own work that we're doing here at the Academy to talk about how we use it. So this is the Vermilion Flycatcher, and it's a beautiful, brightly colored bird. And I became interested in this because a friend of mine, Alvaro Jaramillo, who leads trips to the Galapagos, mentioned that the Galapagos populations of these birds are declining, and they're very difficult to see these days. And in fact, one population on San Cristobal Island appears to be extinct and hasn't been seen for a long, long time. And we were interested in, well, how distinct are these birds? And are there multiple species of Vermilion Flycatcher on the Galapagos, just like mockingbirds? And also, which populations are the nearest relatives? If you look at Vermilion Flycatchers, they actually range from Southern California in the Southern United States through Central America, down to South America, and all the way down into Argentina. And so one of the questions is how have these birds evolved? And so we partnered with a number of other people, and Or Carmi was a graduate student at San Francisco State University who led the work, and we were able to include Galapagos representatives in our phylogenetic work because of the specimens that are in the collection that were over 100 years old. And we sampled birds from 1899 and from 1905 and 1906. And this is the phylogeny that we found. And although we thought that the Galapagos might be most closely related to some of these populations here on the mainland, what we actually found is, and these are the Galapagos birds here, this would be the ancestor of all of the Vermilion Flycatchers. What we found is that the Galapagos birds are sister to all of the mainland birds. And they're so distinct from the mainland birds genetically that they deserve to be separate species. And in fact, When we look on the Galapagos, just at the forms that are on Galapagos, the forms on San Cristobal Island should be their own species, and they had been named their own subspecies, Pyrocephalus rubinus dubius, but we elevated them to a full species. And additionally, we didn't feel that we had enough data at the time to make this split and to know exactly where the split was, but there definitely is information suggesting that the birds on Pinta, Marchena, Santiago, and Santa Cruz are distinct from those on Floriana, Isabella, and Fernandina. So there's probably another split here, but we were very confident in the split between San Cristobal and the rest of the archipelago. But we felt that we would need more data to really figure this out. But now the sad story is that the populations on San Cristobal had not been seen for a very long time and were thought to be extinct. But the information that we published was able to redouble the conservation efforts, bring attention to the Vermilion Flycatchers. And so people started studying them and started really working to figure out what's going on with the Vermilion Flycatcher. And this is the entree for David Anchundia, who's a researcher at the Charles Darwin Foundation and a graduate student at the University of Vienna in Austria. And he is studying the Vermilion Flycatchers. And he's able to get to the field, and he's been surveying the populations on all these islands. And he's been able to collect fresh DNA tissue so that he can continue this genetic work, looking at entire genomes of the Vermilion Flycatchers. And you can see that these islands, the populations are declining fast. And on Floriana, Santa Fe, and San Cristobal, the populations are already believed to be extinct. So the only samples that we have for those populations are still from the Academy's collections. But David was able to get fresh tissues from many of the islands, and he was able to get some eggs from nests that had been abandoned, which gave him enough tissue that he was able to sequence the entire genome. So David came last year to the Academy of Sciences, and he worked for about 3 months on the genetics of Vermilion Flycatchers. And he was able to assemble the entire genome shown here. So each one of these blocks is an entire chromosome, all assembled, and this is the entire genome. And we were able to use this as a reference genome to align all of the other sequences. And so David has entire genome sequenced from about, I think it's about 30 to 40 different Vermilion Flycatchers that he's working to understand the relationships. And we also have the entire mitochondrial genome. And here's some preliminary results that he has building a tree. And again, we were able to show that San Cristobal Vermilion Flycatcher is very distinct from the others. But it turns out that Floriana might be distinct from this group. And these two groups are very distinct from each other. And that there might also be a split between Marchena and Santiago and the others. So this is all work that we're continuing. David has all of the data that he needs, and a lot of the preliminary data analyses are done. So keep an eye out for that. Hopefully in the next year, we'll be publishing a very nice monograph or large paper on the Vermilion Flycatchers. And this will be very important because as they need to or want to renew or rewild some of those islands where populations have been missing or extinct, it'll be important to know which populations to bring them from so that the populations will have the best chance of survival on those islands. There's a lot of other work that we're doing at the Academy too. This is Dr. Jaime Chavez. He's a collaborator and good friend from San Francisco State University. He's an Ecuadorian researcher who's been working in the Galapagos for a long time. And he's been studying evolution in Galapagos finches, but also the ecology and evolution of Galapagos rails using stable isotope analysis, which also relies on our collections. He's been doing a lot of other studies with yellow warblers, and we have several students that we're co-advising, including Pune Kalhori, who's a master's student at San Francisco State, and she's studying climate change in response, climate change response in the endemic Galapagos yellow warbler, and how the populations at higher elevations and along the coast are responding to the changes in climate. And Ezra Mandales, who's studying the relationships of the Galapagos lava heron with the other striated and green herons from around the world. And again, we're able to use a lot of the specimens from the Academy for both of these studies. So in looking at climate change, we're able to look in the past and get DNA information, but also stable isotopes and other information and compare that to modern populations to see how things are changing, and likewise with the striated herons. So in addition to science, though, our new Islands 2030 initiative will be focusing on several archipelagos, including the Galapagos. And of course, there's the science that we'll be doing, and I've already mentioned some of the science that we'll be doing and using the collections, but there's other work that we'll be doing as well. And one of the things that's so important is we want to be, we have all these amazing collections at the Academy, but we want to unlock those. We want to make those available to everyone as much as possible. And so one of the things that we are doing, and we recently got a very nice grant from the National Science Foundation called Galapagos Gateway, which has allowed us to digitize and scan all of the information from some of the larger specimens. So here's an example of a Galapagos tortoise that's 3D scanned, and the resolution of this is very high. So anyone can go and grab these off of Morphosource and study them, take measurements off of those without even coming to the Academy. And likewise, we have other collections at the Academy that need to be worked up. So all of the vertebrate specimens at the Academy are already digitized. The data is online, and now we're doing a lot of 3D scanning and other scanning to make them even more accessible. But some of our other collections are so large that digitizing them is a huge job. And here's a collection of insects that were made in the 1960s on Galapagos. And many of these still haven't been pinned or sorted. So taxonomists have gone through and they've picked out the things that they know best, but then the rest of the specimens still need to be studied. And so one of the things we're working on right now is we just got a huge grant from one of our board members to work some of these specimens up, to digitize as much of the Galapagos collections as possible. And we're hoping to get an IMLS grant to match this other funding that we have in order to also work up these specimens. But in addition to that, we're also doing work with outreach and with communication. So here's our bird and mammal team. This is Mo Flannery from the California Academy working with Gustavo Jimenez from the Charles Darwin Foundation on Galapagos. And they're articulating a specimen of dwarf sperm whale that washed up on the beach in Galapagos. And this is partially for an exhibit, but also partially for science. When I was back in the Galapagos this January, I saw the specimen that we articulated in 2019 in a beautiful case on the public floor of their museum with a lot of beautiful information here. So our teams are working with the teams at the Charles Darwin Foundation to work on museum exhibits and public outreach. Also, iNaturalist trainings. As many of you know, iNaturalist lives at the California Academy of Sciences. And with so many different tourists going and everybody taking photographs on the Galapagos, we're working to train Lindblad expedition leaders and other expedition leaders and tour guides in Galapagos so that they know how to use iNaturalist and how to teach their folks on their tours how to use iNaturalist. And then the information not only helps people identify things in the field, but it also becomes useful data for scientists and park managers in the Galapagos. And we've even helped with a bio blitz a couple of years ago on the Galapagos to teach people how to use iNaturalist and to collect data. So some of the next steps, well, of course, there's the scientific projects and all the different departments that we're going to keep on doing scientific exchanges and information exchanges with exhibits and outreach. We're busy working to develop educational partnerships with the Charles Darwin Research Station and the Galapagos Science Center that's on San Cristobal. And all of the different kinds of things that we can do, media outreach, videos with them. We're planning our Big Bang Gala in 2023, next spring, to be islands focused. So not just focusing on the Galapagos, but many other island archipelagos as well. And we're hoping to be able to fundraise to keep all these projects going and even plan major expeditions in combination with the Charles Darwin Foundation and the Galapagos Science Foundation in the future. So I hope you can see that we've done a lot in the past in Galapagos. We have a lot that we still want to do in Galapagos. I hope that you learned a couple of things about the Galapagos that you didn't know before. And I'm happy to answer any questions that you might have. So thank you very much for joining us today.
Hey, Jack, thank you so much for that. I loved listening. It felt like I was kind of in high school biology again in a really good way, just like relearning how amazing evolution on the Galapagos is. Um, so I'm kind of starting backwards, like from the end of your talk, but what does it mean exactly to digitize collections generally when you're talking like that?
Right. Well, there's different levels of digitizing collections. So the first thing that we usually do is every specimen that we have has a tag with data on it, and it says where it was collected, when it was collected, by whom it was collected, what is it, and that important catalog data is the first thing that we try and digitize and put online. And that way, people all around the world can understand exactly what we have, exactly when it was collected, and get some of the basic measurements and basic data that are on the tag. So that's sort of level one of what we mean by digitizing. And I've already mentioned that all of the vertebrate, in fact, not all of the vertebrate, but all of the birds, mammals, herps have all been digitized. So all that data is up online. You can view it from the Academy catalog or from GBIF and other places that aggregate that kind of data. But then there's also, there's other data that people want too. And so, all of the other, digital lives of specimens. So whenever we do genetic work, all of the DNA sequence data is digital data, and it all lives in GenBank, and that's can all be linked to our catalog data so that anyone searching the catalogs can see that there's genetic data available and look for that online. Additionally, we often will take scans, and especially for things like insects, you can take pictures of the butterflies or the pictures of the beetles. And if they're high enough resolution, those can be used for scientific work. And people from other countries who might have a difficult time coming and seeing them can sort through their own collections and say, oh yeah, this matches the type specimen that's at Cal Academy, or, and there's a lot of work that can be done with that. We have another grant called OVERT, and that's allowing us to do CT scans. So that's just like when we get a CT scan at the hospital, it gives a 3D view inside. And so that makes it possible for like the pickled lizards that we have in the collection. So we have full, they're stored, they were usually fixed in formalin and then stored in ethanol, but if someone wants to study the skeletons or the soft tissue, the muscle, they have to come and do a dissection. And once they do that dissection, then that specimen is dissected, and there's a lot of things you can't do with it. But now with the 3D scans, people can go to this site online called MorphoSource, and they can see the 3D scan and actually get all of the bones measured at very high resolution. All the information is there from the CT scan. So in the OVERT project, and the Galapagos Gateway has actually done a lot of the Galapagos CT scanning. So we have CT scans and 3D surface scans for a lot of the Galapagos material that's been funded by NSF through the Galapagos Gateway. So, and you can imagine, other things too. Wherever there's data, you might want to digitize that data and link it to the collection so that anyone can use that data. So every time somebody publishes a paper, their data is supposed to be made publicly available. And one of the things that we try to do is to capture that data and link it to our collection catalog so that people, when they search our catalog, can find that other data. So kind of a long-winded answer, but that's what we mean by digitize. So there's, a lot of different levels of digitizing.
Yeah, great. Thank you for that explanation. And a question came in. So how has genetic work altered your view of previous Galapagos taxonomy?
Quite a bit, actually. And, of course, with the work with the Vermilion Flycatcher that we've been doing, before they were just, everything that was on Galapagos was considered just varieties of what was on the mainland. And now we know that there's, at least 2, probably 3 different species on the Galapagos. And even within some of those species, there might be more varieties or subspecies that, should be named. And that's important because each one of those groups is, evolved on its particular island and might not do as well on other islands. So if we want to move them around for conservation purposes, we need to know, about its history and what they've evolved to do. And we can really dive into that kind of information with the genetics. And, there's some other great stories from the Galapagos. So, for example, Vicky Friesen's group has been studying petrels on the Galapagos. And there's one island that has, populations that breed during one set of seasons and populations that breed during another set of seasons. But for a long time, they've been thought to be, the same species. But, why do some of them breed this time? it turns out that when she was able to dive into the genetics, that they're actually different species and they're chronologically separated. So instead of being separated in space on different islands, they're actually, this group goes off, flies in this direction and feeds on these resources. Then they come back to breed at this time, and this group goes off and flies off to a different place.
Fascinating.
But because they're feeding on different resources, the breeding time happens at a different time, but they just happen to be breeding on the same island. Wow. And so you would never have been able to figure that out without, the genetics, and then even, sometimes the stable isotope work that people are able to do. So the genetics has been incredibly important. And even so in Darwin's finches, even though, we think we know what's going on, the genetics has told us that there's a lot of interbreeding going on between these different species. And so it's a much more complicated picture than we once thought. And if you look at some of the different islands, we often will call the small-beaked groundfinch the small-beaked groundfinch because of its size, and we define it because of its size. But if you look at the genetics, you realize that those small-beaked groundfinches aren't all each other's closest relatives. And sometimes a different species might have gotten to that island and then become a small-beaked groundfinch. And so that complexity would never, we would never be able to understand that and unravel the history of what's happened without the genetics. Yeah. So the genetics has been really fun because, for the most part, it's told us that we've been right, but those cases that we've been wrong, the story is usually really interesting. So.
Yeah. Oh yeah, that's wonderful. Are there other bird species that you know of that are chronological, have that different kind of adaptation that's based on chronology? Or is this the first instance?
Yeah, that's really one of the only cases that I know of, but the place to look for that would for sure be in shorebirds. But yeah, I mean, that work that work that Vicky Friesen's done is really neat, and I think it kind of blew everybody's mind. That was really fun. Yeah. And a lot of people, the classic idea of how evolution happens is what we call allopatric speciation. Is that when you get to a new island, you're no longer, interbreeding with this other form. So you can each wander down your own evolutionary path, and eventually you'll become different enough to become different species. And so, it might be adaptation, but it might just be, this random walk. And to find out that, the random walk can happen on the same island as long as they're separated by time or separated by the things, and that's called sympatric speciation. And for a long time, people just thought it couldn't happen, but the genetic data has been telling us, no, it can happen, but the times and places that it happens are very special. And so those are really interesting for evolutionary biologists to study.
Yeah, great. And another question is, how is global warming affecting the islands and its wildlife? And does that have anything to do with the Vermilion Flycatchers and their decline?
That's a great question. And there are a lot of things that are happening on Galapagos that are all happening at the same time. So we have climate change, which involves sea level rise. So some of the smaller islands are actually, disappearing or going under the water. And even some of the larger islands, some of the mangrove habitats and some of the coastal, habitats are being very seriously changed by the sea level rise. The different temperatures are affecting things, and you've probably all heard of El Nino cycles. And as those cycles become more and more exaggerated, the droughts become more exaggerated, and the wet seasons become more exaggerated when they do happen every few years. And so that's one of the things that Peter and Rosemary Grant witnessed is one of those the big El Ninos that before that we didn't have as many records of those kinds of things happening, right. And they were able to document those changes. So that definitely stresses the bird populations. And then in addition to, so all those things, the big climate extremes can affect the Galapagos. But in addition to that, with so many people coming, there's a lot of introduced species. So cockroaches, rats, other birds that get introduced to the islands. And all those things are affecting the populations. Plus, some of the islands now, in order to support all the tourism and the food that people need, land is being converted to agriculture. Big towns are popping up, like on San Cristobal. on Santa Cruz Island, and even on Isabella. And, you know, where there's a lot of people, they have a lot of impact on the land, and you know, and convert a lot of the land to agriculture and other things. So so all those things are affecting Galapagos. And in order to, you know, understand what's going on, it's really important that we're able to look back in time as much as we have documentation to try and keep the islands, you know, to their natural state as much as possible. So, and another thing that's really interesting about the Galapagos is that almost everyone's livelihood is really linked to a healthy environment. That's not true everywhere, but tourism, people want to come and see the nature. And so if the nature is lost, you know, that's everyone's livelihood. And so it's a real balancing act. And I think, you know, whether you're a farmer, you're still mostly feeding the people in the town and the tourists who come. You know, all of the different industries kind of centered on tourism. And so, and the science too. So, you know, it's a real balancing act, but I think that there's a lot of incentive in the Galapagos to get it right. And so just like it's a hotbed for studying evolution, it's an amazing place to study conservation, because the different islands each have their own challenges, and they can try different things on different islands and see what works and track the success and the failures. And so, so it's an amazing place to do science of all sorts, but for conservation science as well, it's turning out to be an amazing place to do conservation and management science.
Yeah, thank you. That's so exciting. And then finally, just to kind of to follow that line of thought and to kind of close this presentation is you mentioned about like what about the vermilion flycatchers and potentially rewilding them. So what does that look like? What does that process look like?
Right. So, well, so David is working on all the different challenges that vermilion flycatchers have. And there's also a small fly called Phylornis, which has been introduced to the Galapagos probably around 1960s. And again, we only know about when it was introduced because we have collections at the academies from the 1960s that have Phylornis flies in them. None of the collections prior to that have Phylornis flies. So and if you look at the finches that have been collected over all the ages, you don't see any scarring from the flies, because the flies actually attack the nestling birds, and the ones that do survive are usually scarred around the face. And you begin to see that scarring around the 1960s. So we know that, you know, the Phylornis fly was introduced, but the fly actually, as an adult, feeds on plant matter the way you normally think of flies doing. But when they lay their eggs in bird nests, and the larvae suck the blood of nestling birds, and they crawl into the nose and around the mouth, and then they can kill the birds, you know, if there's enough of them or they eat enough. And, you know, it's rare, but some birds do live through that, and they're usually, you know, very scarred. So one of the biggest threats is trying to understand how to stop the Phylornis flies from killing many of these small birds. And the woodpecker finch is another bird that's affected by Phylornis flies. I mean, almost all the birds are affected by Phylornis flies, but some birds can do better than others. So one of the challenges is the Phylornis fly. And so finding populations that have figured out how to do okay with Phylornis flies might be part of the solution, because there may be some genes for fighting those flies, or recognizing the flies, or picking the flies out of the nest. But the other thing is like there's big work afoot to try and restore the islands of Floriana, maybe Santa Fe, and some of the other smaller islands have already been restored. So they've already done the experiments, and they've been able to eradicate the cats and the goats and the rats and the mice that are on those islands. And so now they're trying a larger island of Floriana. And so one of the things that they want to do is figure out, okay, what mockingbird do we need to put back on Floriana? And they've been able to do genetic work and show that there's populations on Gardner Island, which is basically just a small rock off of Floriana that still has some of the right kind of mockingbirds. And David's work is hoping to identify, you know, which are the closest relatives to the birds on Floriana, so that they can be re-released on Floriana too, once all the predators are gone. And now part of the part of the mix is you want to find the ones that are is most genetically similar to the ones that are on the island, but you also want to say that, oh, the habitat that's available on Floriana looks like this. And so even though the most genetically similar one might be this other bird, if they're in a completely different habitat, they might not be the best choice. So you might be able to look at, well, here's all the ones that are closely related, but here are the ones that are in the same habitat as well. And these populations are doing well, and they might be able to resist phyllornis flies better. So there's a lot that goes into these decisions. But again, every time they do it, it is an experiment. And so each one of these experiments will either succeed or fail, and then they have a chance to redo it again if it does fail. And if it succeeds and everything goes well, then they learn that too. So that's some of the information that goes into the decision-making process for rewilding.
That's incredible. Thank you so much. I actually do have time for one more question. A really interesting one just came in. How do you contain the disease potential from viruses, bacteria, insects, etc., discovered in archive specimens so that they don't escape and infect current species?
Right. No, that's a very good question. And most of the ways that we prepare birds would kill most of those kinds of pathogens. But it is possible that some types of preparation, might not kill them. So for example, today, when we go to a place like the Galapagos or New Guinea or Madagascar, and we bring specimens back to Cal Academy, we have to go through, in fact, you all do this when you go through customs, but there's a part of customs called APHIS, which is the quarantine sort of office. And all of our permits stipulate how we have to prepare our specimens to make sure that we don't bring back any pathogens, and any pathogens are killed. And the things that people are mostly worried about today are things like avian flu, Newcastle's disease, and there's a handful of others, but mostly we can treat those with the different chemicals we use for preservatives or by heating the sample up for a certain amount of time. But then the question is, well, what about those old things? Can we get sick from handling these birds that are in the collection? And no, most of them are prepared in such a way that none of those pathogens would remain alive and viable. But interestingly, the DNA from those pathogens does remain viable, or not viable, but it remains present. And so, for example, in Galapagos, and this is another great use of the collections, there's a pathogen called avian pox that's been released to the Galapagos, invaded the Galapagos, and affects a lot of the small passerine birds. And in order to understand, where it came from and how, Patty Parker and her team came to the Academy's collection, and they looked through literally thousands of specimens of Galapagos birds collected throughout the islands and through time. And they were able to show that prior to 1900, there's no evidence of pox in any of the birds. And between 1900 and 1910, you do find birds with pox, but only on the big islands that have people present. And that's like San Cristobal Island and a couple of the nearby islands. And then after 1910 or so, you can see pox on all the islands. So that suggests that pox probably was introduced sometime at the beginning of the 1900s, and probably on those islands where there were Spaniards. And then you can actually go and sample those lesions and then sequence the DNA from the lesions. And she was able to show that the DNA from the lesions matches the same pox that we have in the Galapagos today, which is related to canary pox. So it was probably brought to the Galapagos in canaries. So one of the hypotheses was it might have been brought in chickens, but my recollection of her work, and I could be wrong about this, but my recollection was that it's not that closely related to the chicken pox. It's not chicken pox, but it's the pox, the avian pox that's found in chickens. But it is more closely related to the canary pox, the avian pox that's found in canaries. So it was probably brought, on ships where people carried canaries. And the Spanish love to carry canaries for a variety of different reasons. But, the canary in the coal mine adage, they were useful birds to have on ships. So a lot of times the pathogen information or pathogen DNA is there. In fact, we've been able to show that taking a little toe pad from a 100-year-old bird, we can test to see if it had avian malaria and detect the DNA from malaria fairly often, so that the old populations, we're getting basically the same kinds of malaria infection rates as we get from the modern populations in islands like New Guinea, where we've done that work. So there's an incredible amount of information, but that malaria that's in those specimens is no longer alive. It's no longer infective, and of course, the mosquito would never bite those specimens, so it would never be able to transmit those. So we think we're fairly safe from all the things that are in the collections. And then things like COVID that everyone's worried about. Those are mostly RNA viruses, and the RNA virus, the RNA doesn't even last in the specimens. The DNA is much more hardy and does remain in the specimens, but the RNA is lost. So those kinds of things only live a matter of, a very short time, days, outside their host. So we think we're pretty safe. But that's a great question. So thanks for asking that.
Yeah. Well, thank you so much, Jack. This has been an absolute pleasure, and I've learned a lot. I'm sure our audience has too. So I'm going to say goodbye, and then I have a few more housekeeping things, but thank you so much, Jack.
Thank you, Christina. And thanks everyone for joining today. Have a great day.
Thanks again. And I'm just here to say that next month, October, is Members Month. You probably have gotten an email about it. Jess, our membership program manager, just dropped a link in the chat. There's more talks coming up, behind the scenes tours on site, special animal feeding. So take a look at the link, and we hope to see you at the Academy soon or back here online. So thank you so much, everybody.